going back downstairs? Do you think? Are you going to come back up? Could you bring me a drink on the thing? Yeah, that's fine. Thank you. Zero. I mean, I've got some in my bag. Priorities. Yeah, come on down. Good evening, everybody. Thank you for coming out to the festival tonight on this beautiful sunny Friday. I'm not going to keep you. I'm just going to hand straight over to Hamish. So enjoy the talk.
Thank you very much indeed. Well, it's very nice to see all of you out this evening. I was told just as we started this is going to be recorded and available in a few weeks' time, so you could actually have just stayed in the pub and not bothered to come here at all and heard the same thing. So I have you now captive for the next 2 or 3 hours. If you have come to this talk looking for gardening tips and how to grow your vegetables better and things like that, you will be sorely disappointed. I am a plant scientist. I'm particularly interested in the evolution of flowers and the evolution of plants. And what I'm going to be telling you tonight is plant science in the context of gardening. Why do plants behave as they do? Why does all that stuff you do in the garden work? What's the science behind it? And what's going on there. Now, I'm loosely going to be hanging all of this on the gardening year as we go through from germination through flowering, seed set, and to winter as well. So we're sort of looking through from about January through to about the end of the year. No, I did introduce myself. That's fine. I'll edit that out in the cut. That's fine. Okay, so let's start by having a look at seeds and when it's time for those seeds to wake up. So how does a seed know that it's the right time to germinate and come out from underneath the ground? Some seeds do sit on the ground, on the surface of the soil, and so they would be able to sense light. But many seeds, and particularly the ones you're planting, you put a hole in the ground, you put the seed in the hole, and you cover it up. So it can't be light. So it's got to be something else. And there's lots of risks for this seed to be getting it wrong. If it comes up at the wrong time, it could be too cold, there would be frost, so that would be death, which is obviously bad. There might be a lack of pollinators, because the insects which need to be pollinating the flower aren't out yet. It might be a lack of other plants of the same species, so it couldn't pollinate, so there wouldn't be anything to pollinate with. Maybe there wouldn't be enough time for it to grow if it came up too late, the death of the fruit over winter and so on. So getting germination time right is really very much of importance. And something which is worth noting, which I often forget, is that seeds are alive. They're living things. It's just that their metabolism has slowed down compared to the plant they came off. The metabolism is still going on. So there's biological processes going on, and it's those biological processes which are telling it when to germinate. So let's have a look inside a seed. This is a ginkgo seed, which we've cut in half here. And a seed is an embryo. That's the two little... have I got a pointer on here I can use? Oh, not that one. There we go. How do I show the pointer? I don't know how to show the pointer. I will just tell you what's going on. You've got these two little leaves up there, and down at the bottom of it, you've got the embryonic root in there as well, and where this plant is going to grow from. And the rest of the seed is nutrition. It's called the endosperm. It's there to provide nutrition to the developing shoot before it can photosynthesize, while it's pushing its way up from the ground. You might have seen this in a peanut. Most of the peanut is the same as that food for it, and the little nubble that sometimes falls off, that's the embryo. So seeds are dormant. They're not dead, they're dormant. They're waiting for the right time to come up. And there's a couple of different ways of providing that dormancy. It could be physical dormancy, so it could have a hard coating over the outside of it, and the embryo physically can't get out of that hard coating before it's been broken down some way. And that some way could be by cold temperatures, so frost could shatter it, or by fire, or by freezing or drying out, or maybe even by digestion, so going through the digestive tract of an animal and the chemicals there breaking it down as well. Also, the tissue surrounding the embryo might not allow gases in, it might not allow oxygen in or carbon dioxide out, so there may be this barrier there to the embryo growing along. There could also be chemical dormancy. And chemical dormancy is where the seed coat or the seed prevents the embryo from developing. So maybe there's an inhibitor in there which breaks down over time. It's interesting to think about this in the context of plants which, biannual plants, so things like a foxglove, which starts to grow in one year, remains dormant as a plant over winter, and then develops the flowers the next year. In that case, the flower buds are dormant rather than the seeds. So how might this dormancy work? How might it be measured? How might it be broken? So one way of thinking about things is with temperature. Here I've got a graph where the vertical axis is temperature and the horizontal axis is time, and we're thinking about this over several months. So we're thinking like starting in the middle of winter. So temperature is very low. And then let's say maybe you have a little bump in January of a slightly higher temperature, goes back down again to cold in February, and then heats up again in March time. And when the temperature goes above that dotted line, the trigger there, that is when the plant decides, when it's triggered to germinate, when the seed is triggered to germinate. But what happens if, as we've had so recently, we have a much warmer spike in the middle of January? In that case, the temperature is going above this trigger level, so you might think that the seed would trigger its coming out of dormancy there. And sometimes that does happen with some species, but there are clever ways around it. And we can use a trigger protein which is created when the temperature gets high enough, but which is degraded at a slightly slower rate over time. Let's have a look at how that works. So I've still got temperature on there, but the dotted line is now the level of the trigger protein, above which the trigger protein needs to go. And the trigger protein is made when it gets hot, but degraded all of the rest of the time. Let's have a look what happens there. I'm showing this in pink. When the temperature is low, none is made. When the temperature kicks up, a bit is made, slowly, but when the temperature drops, it's degraded again. And then when the temperature rises again and stays hot, the trigger protein is then made. It doesn't break down because more is being made to top it up, and eventually it goes over the level which it needs to go at. So that's a clever way of being able to measure a length of time simply by using this trigger protein as a timer. But, I hear you cry, seeds are made in August or September when it's warmer. Why don't the seeds germinate then? And so there's going to be something. So I've got another graph here with the temperature as if it were at the end of the year, and we've still got a hot January. So now what I'm going to introduce is a suppressor protein, which is made by the parent plant and put into the seeds. It gradually decays over a few months. And while that suppressor protein is there, the seed cannot germinate. Let's have a look at what that is. The suppressor is now in blue, and the trigger protein is in pink. So we do start out, there we go. The suppressor protein is there, the trigger protein can't be made. The suppressor protein is gradually removed, and then, as before, we measure the length of time that it's been warm, and only when it's been warm enough for long enough does the seed germinate. Now, this is relatively theoretical. There are lots of different examples, and it's quite a lot more complicated because there's so many different signals which can be integrated. But this is the sort of way that plants can measure time, and particularly measuring time over the course of months. Okay, so we've germinated. Now, how does the plant know which way to go? Here we have a sunflower seed, which is very happily growing. This is a time lapse. The sunflower seed is growing upwards, the root is growing downwards. This seed is doing exactly what it should do. But how does it know which way is up? Bulbs do this too. If you plant a bulb, it will come up. If you plant a bulb upside down, the shoot will come out, and then it will curve round and come up. And this bulb will flower a couple of weeks later than if you put it in the right way up, as my father found out when he planted some bulbs for my wife and I wedding, and he planted them upside down to make them just that little bit later, which I thought was quite clever. So the plant needs to measure gravity. It needs to know which way is down and which way is up. It can't do that with light because it's under the ground. So and the way that we know that plants measure gravity is from an experiment back in the 1870s with a fabulous piece of kit where they got a plant pot with a seed in it, and they put it horizontally and rotated it. And the light was constant, and the plant came out sideways, because the plant didn't know which way was up, because it was kept on being rotated. And so instead it grew sideways, because there was no up, no down, and the roots grew back into the pot. So that's how we know that plants can do it. How do they do it? If we look at an iPhone, I hate this thing, it was very cheap. If we look at an iPhone, what we have is a small box with a ball in the middle of it, suspended by 6 springs. And when you move the phone around, that ball joggles around within those 6 springs, and the forces on those springs tell it which way it's going and how much it's accelerating. On cheaper, older digital cameras, you've got a thing like on the right, where you've got 4 plates and a moving ball. And when it's this way up, the ball connects these 2 plates. When it's sideways, the ball connects those 2 plates, and that's how those cameras detect orientation. And actually how plants detect orientation is a very similar method to that one. Here is a cell, and inside a cell there is a sort of watery soup called cytoplasm. It's quite thick. And inside that are all of the things that make the cell work, like the nucleus. But something else which is inside there are little organs called statoliths. They're in blue here. And those statoliths are made of starch, and they are heavier than the cytoplasm. So what will happen over the course of a few minutes is they will sink to the bottom of the cell. If we turn the cell on its side, they'll sink again to the new bottom of the cell. So that's the sensor. That's how it knows which way is down. Bank that thought. We'll come back to how it knows how to grow in the right direction in a second. So now I want to move on to having a look at something which I think probably most of you will know if you're gardeners, which is pinching out and pruning. On the left here, I've got a sweet pea plant. And the allotment holders next to us will always tell us when you see 2 or 3 leaves, pinch out the growing tip, and that will make your sweet pea plants nice and bushy. Likewise, we need to be cutting plants. I think this is a grapevine on the right here. When we're pruning them, we cut just above a bud, and that will stimulate the bud to start growing where it wasn't growing before. But how does the plant know that now is the right time to make those buds grow? What's the sensor there? What's the mechanism behind it? How does a plant know it's got a growing tip? And here I have a cartoon of a little plant with new leaves at the top and older leaves down at the bottom. To understand this, I'm going to introduce you to a plant hormone which is called auxin. Auxin does basically everything. I was talking to my boss about this earlier today, and she said it's worth thinking of it like information. Auxin tells a plant about its surroundings. It's used in almost so many different biological processes within plants. It's sometimes difficult to understand how it does everything, but it's all about context. Auxin is produced in young leaves and right in the growing tip of the plant, and it's transported rootward through the vessels within the plant. So it's made at the top and it's transported downwards. This is how the plant knows it has a growing tip, because auxin is being made. It's not made in any of those other leaves, and it's not made in the little buds in the sides of the leaves as well. But how does that work? Here are some cells. I've got 3 cells in a row. Auxin is made at the tip, and you can see there's these little red blobs around the outside of it. These are proteins embedded in the cell membrane, and they're called pin proteins. These are transporters of auxin. They transport auxin out of one cell. So if auxin is being made at the top, The pin proteins localize where there is high auxin, so they localize at the base of the cell because the auxin is coming through the cell, and they then transport that auxin into the next cell, where pin proteins localize at the bottom, transport it into the next cell, and so on. And what we end up with is a canal of auxin coming from the top of the plant, going all the way down through the roots. When it passes a bud, I've got this bud as 2 cells on the right-hand side here. We've got this canal of auxin going down. And that canal interacts with other hormones and other proteins as well, and it stops growth of the bud. The bud is not able to grow. It's not able to produce its own auxin and start and join that canal because the canal has essentially walls on the side of it. It won't let any more auxin from any other branches join in. So then we cut the top off. There's no more auxin coming from the top. The canal is broken. And that means that the bud can be triggered to start growing, and it can join that canal or make a new one of its own back down to the roots. So that's how a bud knows that there is a growing tip, and once we've cut it off, the bud knows that it can start growing. There's even more interesting stuff going on here because it's usually only the top bud that starts growing. It's not the second one or third one or fourth one down. And that's because the roots are also sending signals back up saying you are close to the root. You have this gradient going on there. And so buds which are closer to the roots get inhibited, and buds which are further away are more likely to start growing. You might also see this in seed potatoes. If you have a seed potato, usually only one of the eyes will start growing. But if you cut the seed potato up into pieces, they will all start growing. And it's because when one of them is growing, auxin is telling all of the other ones, don't grow, we've already got a growing tip there. Back to gravity. You remember I said bank that thought with the statoliths which had fallen to the bottom of the plant. Let's have a look at how that plant grows. Here I have on the right a small shoot with a root and a couple of leaves. If we turn that on its side, the statoliths then all fall to the bottom of the cells within that plant. And it turns out that statoliths trigger the accumulation of those pin proteins I was talking about, and they co-localize, they go to the same place. And that drives a higher concentration of auxin on the bottom of the shoot, so when we've tipped it over, on the bottom of the shoot than on the top. And auxin then triggers growth. In the shoot, auxin makes it grow, so it will grow faster on the bottom than on the top, and it will curve upwards. In the roots, it does exactly the opposite. Auxin slows down growth. So on the side, if we've got an accumulation of auxin on the bottom and not on the top, it starts to curve downwards. And that is exactly what we see when my clicker works. And the plant grows like that, so that it knows it's on its side and it grows away. So that's how we integrate the gravity signal into a growth signal. While I'm here, let's have a brief diversion into grasses. We've had a look at that growing tip and auxin and things like that. And I just want to mention grasses because they're really quite cool. On the left, we've got a picture of a slightly tall grass. Maybe it's maize, maybe it's a grass like out in a meadow. And you see we've got a growing stem with leaves coming off it every so often. It's like most other plants. We've still got a growing tip in there. But I want to look at the type of grass which we grow on our lawns, which is the one on the right. In that, the spaces between the leaves have become compressed. So there's no space at all between those leaves. We've just got one coming off that way, then that way, then that way, then that way. And they grow upwards. And that's a really important thing to know. We've got the growing tip down the bottom, and these leaves grow upwards. And they grow from the bottom. They don't grow from the tip because it's not a growing plant. The zone of elongation within those leaves is right down at the bottom. And that means that when you mow it, you're not cutting off the bit that's growing. You're cutting off the bit that's already grown. So you're not disrupting the growing tip. We're not pinching it out like in the plant I showed you before, or the sweet peas. Instead, you're cutting off the bit that's already grown. On your lawns, you'll do that with a lawnmower. If you have a horse, then the horse does exactly the same thing. It's cutting off the bits that have already grown, and it's leaving behind the bit that is growing. And I think that human civilization is underpinned by this bit of plant anatomy. Because if the growing tip was being cut off by your horse, then your horse would eat all of the grass that was around. And if you think how long it takes a sweet pea to start growing up and bushing up, it takes quite a long time. Whereas if you think how fast your grass grows once you've mown it, so because you're not cutting off that growing tip, it will grow back nice and quickly and provide more food for your animals. That means if you are somebody around 15,000 years ago, you can put your horse in a field and then move it to that field and then move it back to there. You can stay in the same place. You don't need to be moving around to continually find forage for your animals. And if you don't need to move around to find forage for your animals, you can start building a city and civilization. So it's this single bit of development of a single plant, of a handful of plant species, which, like I say, I think underpins the entire of human civilization, which I find quite cool. Okay, brief drink, and then fertilizers. I'm not going to drink fertilizer. That came out wrong. Okay, fertilizers. There we go. Plants will grow relatively happily within soils, but as we found for our crops, we need to be adding nitrogen, phosphorus, and potassium to the ground, and that will help them grow better. And that's what NPK means in this context: nitrogen, phosphorus, and potassium. Production of nitrogen fertilizer happens at a staggeringly vast scale. I hadn't realized quite how much before I started writing this talk. It's made from nitrogen in the air, plus water and natural gas. So the stuff to make it is pretty cheap and relatively available. But we make it at an enormous scale. 2% of global energy is used to make nitrogen fertilizers. And then more is used to ship it around the world. It's enormous. And then we dump it on fields. And only about a third of it is taken up by the plants. The rest of it is washed away. So, about 1.4% of global energy is used to produce something which is just washed away. Phosphorus isn't something we can get from the air. Phosphorus we have to dig up. It is beginning to run out a bit, although I think the numbers are not quite as bad as the newspapers sometimes make out. There's a statistic about 400 years, which is often quoted, but it's not actually necessarily true. But once we've got this phosphorus rock, it's treated with acid to make something which the plants can use, they can take up. And again, we dump a lot of it on the ground, and only 20% of that is taken up by the plants, and the rest of it washes off into rivers and soils and things like that. That is an awful lot of wastage, and it also causes a lot of problems in rivers. The good news is potassium's pretty much fine. We're okay on potassium, so I'm not going to talk too much about potassium. Instead, I'm going to talk about phosphorus and nitrogen. I'll do phosphorus first. Plants are not very good at taking up phosphorus, and the soil particles hang on to phosphorus very tightly. They bind phosphorus really very well. So even if it were available, even if plants were good at taking it up, it's not actually available so much. But the good thing is they have some help, and they have some help from arbuscular mycorrhizal fungi. These are fungi which you might have heard of. You can buy them as what's called root grow, or little granules which you sprinkle in along with whatever it is that you're planting. And these fungi are very good at taking up phosphorus. And they also grow an enormous, it's called a mycelium, a net of fungal tissue throughout the soil. So they're massively increasing the surface area which is available to take up that phosphate. And what they do with plants is very clever. They'll come along in the brown bit at the top, which is the soil. You get this little, it's called a hypha, coming along. And when it senses a plant, there's a slightly complicated handshake that goes on. And then it will form a sort of a lump on top of the root. And a very high pressure peg will come out of the fungus and penetrate through the cells of the root of the plant. It doesn't break the cells, it goes between them. And it penetrates right into the middle of the root till it gets to the right place. And then it penetrates the cell wall of a cell and forms this little tree-like structure which is called an arbuscule. Again, it doesn't break the cell. It's like a hand in a glove. So this gives a really big surface area for the root to talk to the fungus. And the fungus gives the plant phosphorus, which it has collected with its mycelial mat. And in return, the plant gives the fungus carbon, which it has fixed from the air through photosynthesis. So it's a mutualistic relationship. The fungus benefits, the plant benefits. But this is a way that the plant is able to take up a lot more phosphorus. Let's have a look at nitrogen. You might know about this one. These are root nodules in a legume, so in a member of the bean family. And these are bacteria. They're of the Rhizobia genus. Again, there's a complicated handshake between the plant root and the bacteria, and it welcomes them in, forms these little nodules, giving the bacteria a place to live and grow. And the bacteria fix nitrogen from the air, turn it into ammonia, and give that back to the plant. So the plant gives the bacteria somewhere to live and reproduce, and in return, the bacteria give the plant nitrogen. So this is a way of taking up more nitrogen from the soil. So how could this have evolved? Either of these things. It seems a staggeringly unlikely thing to be happening, this really complicated set of stuff that was going on. And it's probably best to think about it in terms of what fungi and bacteria usually do, which is infect plants and cause, or infect things in general, and cause illnesses. So what would probably have happened before all this is that a fungus came along and infected a plant and made it sick and possibly eventually killed it. But once, a fungus came along where there was a mutation in it. And instead of killing the plant, it leaked phosphorus. And the plant, for which it did that, benefited and grew better, set more seed. The fungus, maybe the fungus grew better because the plant was leaking some carbon through its roots, or maybe the fungus did actually penetrate in the first place. I'm not really sure. But it starts out from these accidents of interaction, and then whenever there is a small benefit to either of the organisms involved, they're selected for, they're able to reproduce better, make more seeds, make more fungus. And so through that, that's how evolution works, these tiny little incremental changes. It's the same with the bacteria. There would have been this handshake with the bacterium trying to infect or kill the plant, and something went wrong somewhere. And it went wrong in a fabulous way for a plant. The interesting thing with this, though, is that with our vascular mycorrhizal fungi, not all plants do it. In particular, the brassicas don't do it. So you can shove as much root grow as you want, along with your seeds for cabbage and things like that. It won't make a blind bit of difference, because the plant doesn't know how to do that handshake. Rhizobia bacteria, you only really find them in legumes. So again, we don't get root nodules like that in wheat or rice, for example, or lots of the crops where we do dump all of this phosphorus and nitrogen on the soil. If we can genetically engineer those crops so that they can do that handshake, so that they can welcome the bacteria or the fungus in, we will be able to massively, like a step change, the way that we produce our crops and the amount of crops that we're able to produce for the amount of fertilizer that we put on. And if we remember back to the idea that, 1.4% of world's energy is essentially wasted on fertilizer, that's an awful lot of energy which we could be using for other things. Okay, so we've had a bit of a look at how plants grow. Now I want to have a look at how plants protect themselves from predators. And my colleagues in zoology tell me plants are much less interesting than animals, because when they're faced with a dangerous situation, they can't get up and run away. They can't do behavior. I tell my colleagues in zoology that plants are massively more interesting than animals, because when they're faced with a threat, they can't get up and run away, they can't do behavior, they have to work out other ways of doing it. So I'm going to show you some of those now. And the first I want to show you is poison. This is my favourite part of the talk, I just love this bit. This is bracken. This is the most widely distributed fern in the world. And bracken makes cyanide, which poisons animals which eat it, which is why, when you see on the fells and things like that, you see basically well-grazed grass and bracken. That's because the sheep are clever enough to know that they shouldn't eat the bracken. And if a sheep knows that it shouldn't do it, then the plant is definitely onto something. Cyanide works by poisoning mitochondria, which are things in cells a little bit like the statoliths, which I was talking about. And they make energy from glucose. They're the bit which generates energy within the cell. They're often called the powerhouses of the cell. So cyanide disrupts the processes which are used to make that energy. But, I hear you cry, bracken has mitochondria in it as well. How can it make cyanide? How can it make something which is going to poison itself? And that's where plants are amazing, because many plant cells have vacuoles, which is a compartment within the cell full of water and a bunch of other stuff. And in this case, it's full of, as well as the water it's got in it, a precursor to cyanide. It's not cyanide, it's totally harmless, but it can be broken down into cyanide. And the thing that breaks it down into cyanide is stored by the cell in the cytoplasm, not the vacuole. So it's compartmentalized it. It's keeping the enzyme which breaks the precursor down well away from the precursor. And that means that we have happy bracken growing wherever it wants to. But then a sheep comes along and it eats it. And that chews up the cells. It mixes the contents of the vacuoles with the contents of the cytoplasm. It means the precursor is degenerated into cyanide, and that's where the cyanide comes from. And then it kills the sheep or makes it somewhat unhappy. So it's a really clever way of keeping the poison until it's needed. Almonds used to do this as well. So the almonds that we eat, wild relatives of almonds also do exactly the same method, compartmentalizing the precursor and the enzyme as well. But now we have bred out the cyanide because people do not, for some reason, want toxic marzipan. Okay, let's have a look at another poison. This is on tobacco. Now, the poison that you'll know about in tobacco is nicotine. It's obviously used by us as a relaxant in small quantities. But I'm not going to talk about nicotine in this one. What I'm going to show you here is a protein which tobacco makes. And this protein is an antidigestive compound. It means that caterpillars which eat it are not properly able to digest food. And you might think that that means that they starve and die. But that's not, it seems, how the tobacco kill the caterpillars which are eating it. So how do they do it? Here is a video of what happens when we poke a caterpillar which is feeding on tobacco which has been genetically engineered to not make this toxin. So this is tobacco which is safe for the caterpillars to eat. Let's have a look at what happens here. They're going to poke this caterpillar, and the caterpillar twitches and says, "Oi, get off." If we poke it again, a bit more of a wiggle, the caterpillar sort of starts to move away a bit from the thing. It's not an especially happy caterpillar. And if we pick it up, it starts to wriggle. Now imagine if that's not tweezers, but a bird. The bird comes along and tries to start eating the caterpillar wriggles, and actually it's quite likely to get away or fall out of the bird's beak or surprise the bird in such a way that it has a slightly higher chance of surviving than if it didn't do that wriggling. Let's now have a look at what happens on a different, there we go, on wild type tobacco. This is tobacco that does make that toxin. Let's do exactly the same thing. If we poke that caterpillar, it doesn't mind. It's pretty happy there. And then if we pick up the caterpillar, it just sort of allows itself to be picked up, really. Come on. Oh, it's changing for the tweezers. There we go. So the antidigestive doesn't kill the caterpillar out of hunger. It relaxes it to death by making it so relaxed that it just doesn't care if a bird is coming along as well. Nicotine does also affect insects in a very similar way to how it affects us. It interferes with their nerve communications. And you might have heard of neonicotinoid pesticides. They're always in the news about the bee-killing pesticides. I actually think that's a terrible way of thinking about it. I don't like calling them pesticides because it makes it sound like they are only killing the pests that we want and not killing the good insects. They're insecticides. They kill insects. It's what they're supposed to do. So I prefer calling them neonicotinoid insecticides. And they're amazing at doing what they do. You need a very, very, very tiny amount of them. They're absorbed into the plant, they travel through the plant, and anything which eats the plant takes up this insecticide and is killed. And when they were developed, the nectar and the pollen was tested, because we know that it goes through the plant. And the nectar and pollen was tested, and we couldn't find any of the residues of these insecticides in the nectar or the pollen. And so we thought, that's fabulous. It's not put in there. It's only in the leaves of the plant. This is okay. But actually, it turns out that our instruments weren't sensitive enough. And it is in the nectar and the pollen at parts per billion level, so like 1 part per billion. And that's enough to affect the brain chemistry and the brain signals of navigating bees and disrupt how they learn. So that's why we thought they were okay, and then further testing proved they're not. So now they're banned, which is good, isn't it? The trouble is, we do need to use some insecticides. We still need to protect our crops from the things that are eating them. So actually what we've had to do is go back to other things which are more harmful in different ways. And it's always interesting when there's a headline that the government has approved the banned bee-killing pesticides. I went and had a look at the regulations governing them. It's usually only used in sugar beet. It's sometimes authorised to protect sugar beet crops against aphid pests, and the way that it's applied is as a seed coating. So there is a coating on the seeds when they're planted, so the developing seedling absorbs this insecticide from the seed coating. It's not sprayed on, only used as a seed coating. And that's because the seedlings of the young plants are particularly vulnerable to aphids. Aphids transmit viral diseases. It's called viral yellows, and it can reduce the sugar beet crop by up to 50%. So it's a devastating pest or virus for sugar beet farmers. The authorization criterion for using it is an independent virus prediction report of above 65% yield loss. So it's not always used, it's only used when the yield is predicted to be very low. It's not sprayed, like I said, it's only used at the time that sugar beet is planted. Sugar beet isn't insect pollinated, so insects don't go and visit it. And after the sugar beet has been harvested, only non-flowering crops are allowed to be grown for 32 months after harvest. And also, there is a requirement to prevent flowering weeds in this soil for 32 months as well. So there are an awful lot of safeguards built in to the use of these pesticides. And there obviously have been reports on the risk of these pesticides spreading into field margins. And actually, a study concluded that significant migration into field margins following application of a pelleted seed treatment would not be expected. So it's very easy to jump on the we must stop using the bee-killing pesticides bandwagon. But it's always worth going back and having a bit more of a look about the regulations governing it. Farmers aren't stupid, and they want to preserve the land as best they can. I know I've talked to lots of them, and often they're coming at things from the same point of view as the rest of us as well. It's just interesting to go and have a look at that sometime. Anyway, moving on. More poisons. I talked about proteins being used as a poison. We can also make poisons using single amino acids. And amino acids are the building blocks of proteins in every living organism. There are 20 amino acids which are used naturally in the world. We have 20 amino acids which make up our proteins. Plants have 20 amino acids and so on. And one of those amino acids is alanine, which you can see on the top there. The acid bit is the OOH on the right, the amino bit is NH2, and then on the left-hand side you've got H3C. This is the side chain of the amino acid. This is the bit that does chemistry. And on the bottom, we have another amino acid which isn't found in natural proteins. This one's called beta-cyanoalanine. It's still got the OOH, it's still got the NH2, but then there's a triple bond N in there as well. And that triple bond N as a side chain means that it does chemistry differently. The plant on the right, common vetch, makes this beta-cyanoalanine and hides it away so it can't be used by the plant itself. And then when an animal comes along, it will eat this beta-cyanoalanine. That will be absorbed into its bloodstream, and the animal will use that amino acid to make proteins within its own body. And because the chemistry of that side chain is different, it messes up all of the biological processes in which which are caused by the protein in which it's been incorporated. And that means convulsions, death for the animal which is using it. This one's really quite toxic. I worked it out. I would need to eat 12 grams of that amino acid for it to kill me. So it's a really clever way of poisoning an animal without having to make a specific complicated poison. Here are two of my favourites. Hormones, as I mentioned auxin already, animals use hormones as well. They're produced in parts of our brain and various different places and transported around our body in the blood. And one hormone, which you'll probably have heard of, is progesterone, the female sex hormone. Yams, wild yams, which are a relative of the morning glory, they make diosgenin, I think it's called, which is a precursor to progesterone, the female sex hormone. And it can be broken down into progesterones and estrogens, and it also affects the level of estrogens within the female animal. This is acting as a poison, but over a long term. The plant is playing the long game. So an animal comes along and eats it, female animal comes along and eats it, and it acts as a contraceptive. It stops the animal ovulating. Oh my goodness me, there are so many people here. This is terrifying. I thought I was on my own, practicing. So the female animal comes along, eats this yam. It acts as a long-term contraceptive. Animals which eat this produce fewer offspring. And then those offspring come along and eat the yams, and they produce fewer offspring, and eventually the population is depressed to a level that they die out, or they stop eating the yams. So it's a really clever way of messing up animal fertility. And actually, the hormone which is made by the yams is more potent than the hormone which is made by the animal's body themselves. Super cool. But I think this is my favourite one. An animal hormone called ecdysone. Now, insects and other arthropods need to shed their skin in order to grow. They're not like us with bones on the inside. They have an exoskeleton. And so they will shed their skin when it's the right time. There's a soft version of the animal inside which expands and then hardens off. And that's how arthropods are able to grow while still having an exoskeleton. And the process of molting is mediated by a hormone called ecdysone. You can possibly see where we're going here. The plant makes ecdysone. An insect comes along and eats it. It then gets higher levels of ecdysone throughout its body. And its cells go, oh, right, okay, time to shed my skin. But there's no cells underneath it which are ready to harden off and expand. So the animal literally falls apart because of what the plant has done to it. I think that's fabulous. That's way better than animal behavior. So that's plants defending themselves against animals. What about defending themselves against other plants? To tell you about that, I'll tell you about coffee. This coffee is a plant that makes weed killers. And this weed killer is in the form of caffeine. Now, you know that as a stimulant. And actually, caffeine does act. They make caffeine in their nectar. And that does act to change bee behavior and make it so that bees learn faster and are more constant in coming back to the flowers as well. That's some really cool research there. But the caffeine is also a weed killer. It's in the leaves of the plant. And when the leaves fall off and fall to the ground and decompose, they release caffeine into the soil. And that prevents the germination of other seeds and seedlings around the plant. And it's how the coffee plant eliminates its competition. So you'll have a coffee plant and there'll be a barren area around it because the plants, the little seedlings which are growing, are not able to germinate because the caffeine is inhibiting them. And you can then think there's a massive selective pressure for the little seedlings to start developing resistance to caffeine. And then there's a selective pressure on the coffee tree to develop different versions of caffeine or better better weed killers to better fend off those plants. You'll find evolution is always full of these arms races where one thing is trying to benefit itself, something else is trying to benefit itself as well. So that's natural stuff. What about the weed killers that we use? Here is what happens when you spread weed killer on a golf course thinking it's fertilizer. That was in the news a couple of years ago. It's slightly embarrassing. Weed killers kill plants, obviously. And there's a couple of different weed killers I want to briefly talk about. The first is Roundup. And Roundup is a non-specific weed killer. It just kills plants. And the way that it does that is by targeting a protein within plants which makes amino acids, makes those building blocks of proteins I was talking about earlier. Plants need to make those amino acids in order to be able to grow. And Roundup disrupts the protein which makes 3 of them. So it depletes that plant entirely of those amino acids. They all get mopped up into proteins. And then it can't make the proteins it needs to live, and it will die. So that's how Roundup works. Of course, there are now some Roundup-resistant weeds, and those Roundup-resistant weeds have developed a slightly different version of that protein which makes those amino acids because there was an enormous selective pressure for them to do so. So that's when we need to start developing new weed killers ourselves. What about broadleaf-specific weed killers then? These are weed killers you can spread on your lawn and they will kill off the broadleafed weeds, but they won't kill the grass. It's really clever. And actually what that is a version of something I've already told you about. It's a version of auxin. And broadleaf plants will take up weed killers like this. There's a particular one called 2,4-D, which is very good and also horribly toxic to people. It was part of Agent Orange, I think, in Vietnam. But 2,4-D is absorbed by broadleaf plants really well and transported through them really well. And it's not absorbed by grasses at all well, and it's not transported through grasses at all well either, for reasons which remain elusive. And what it does in the broadleaf plants is cause uncontrolled growth. You might have seen it when you spray this, when you spray broadleaf-specific weed killer on them. They all spall up and start to twist. And that's because growth is disrupted. It's all growing in different places. And eventually the plant will die off. There are other things we could use as general weed killers. I don't have time to look at all of them, but some of them, for example, target chloroplasts. So the green organs within plant leaves which do photosynthesis. We don't have chloroplasts, animals don't have chloroplasts, so these things wouldn't be effective against them, they're only effective against plants. And malaria, because it turns out that the malaria parasite evolved a long time ago from a photosynthetic alga, and the malaria parasite still has inside it a remnant chloroplast called an apicoplast. It doesn't photosynthesize now, but it's still there, and it's still needed by the parasite to be able to live. So actually, and this is by no means medical advice, some weed killers are able to target the malaria parasite and stop it in its tracks. It's absolutely fabulous. And I used to work in a lab where they worked on plants and malaria, which is a very confusing conjunction. So how does a plant know when to flower? We've, assuming it's grown happily and everything like that, it needs to flower at the right time. Because if it doesn't flower at the right time, well, we've got all of the problems which we had before about coming up at the right time. Your pollinator isn't there. Another plant which you can pollinate with isn't there, and so on. Now, perhaps it's measuring the amount of time since it germinated, but it can't be that, because if we've got a perennial, a rosebush or a cistus or something like that, that keeps on flowering year after year. So it can't be measuring a certain time after germinating. And actually what it might be measuring is the time of the year. How does it measure that? What's the thing that mostly changes throughout the year, particularly in the, away from the equator? It's the length of the day. So the way that several plants know when to flower is by measuring the length of the day. There's a couple of different types of plants. There's plants which flower when the days get longer than a certain threshold or get shorter than a certain threshold. I'm going to look at the latter one. They're called short day plants. Here I've got a graph with 4 sections on it. There's a yellow and a black section, and this is representative of a day cycle. So we've got the yellow is light, the grey is night. And during the day, our old friend, the trigger protein, it's a different one, but it's a similar sort of thing. During the day, that trigger protein is made pretty quickly, and then during the night, that trigger protein is broken down. This trigger protein is an inhibitor of flowering, so when it's present, you cannot flower. And when the nights are this short, the trigger protein is not broken down fast enough to get down to a 0 level. If we increase the length of the night, the trigger protein gets broken down at the same rate, but eventually all of it is broken down before night ends. That removes that inhibition and flowering can start. So actually, well, how do we know that the plant is measuring the length of the night and not measuring the length of the day? There's a beautifully elegant experiment here. I've got that bottom graph now on the top. And what some researchers did is they gave the plant a little flash of light in the middle of the night, only 5 minutes. But that's enough for the plant to make more of that inhibitor protein. And so what happens then is plants which are subjected to this treatment don't flower. Now, chrysanthemums need long nights in order to be able to flower. And we can obviously buy chrysanthemums in the spring when the days are long. So nurseries are using artificial lighting to induce flowering for cut flowers. They will have them in entirely blacked out greenhouses and control the light and control the dark so that chrysanthemums are induced to flower when we want them to rather than when they want to. Feels like sounds like I'm gliding. We also get this with poinsettia. So this is a plant you may have seen at Christmas, and you've got these lovely red bracts on it. But if you keep it until next year, it looks a bit miserable. And that's because the production of those red bracts is based around having a long night. And what you do with your poinsettia is you put it in pride of place on the kitchen table and then keep the lights on until you go to bed at 11 o'clock. It needs a long night. So if from October you take your poinsettia and you put it in a dark room or a cupboard or something like that, where it won't be exposed to any light from 5 o'clock in the evening till 8 o'clock in the morning, then it will start looking happy again for Christmas. Possibly the most useful part of the entire talk. Okay, now we've decided when to flower. Let's have a look at how to make a flower. For those of you who did GCSE or O-level a very long time ago, here's what a flower looks like. Going up from the bottom, we've got the stem, then we've got the sepals, which are the things which enclose the bud. We've got petals, which are petals. Then we've got the stamens, the male bits, which have pollen on the top. And then we've got the carpel in the middle. That's the female bit. And the carpel is divided to stigma at the top, style, which is the bit which supports the stigma, and then the ovary down the bottom, which is where the egg is. Just remember the structure of that female bit, stigma, style, ovary. We'll come back to that in a moment. And also remember where the egg is. It's in the ovary down at the bottom. So how are these flowers made? You remember I've said we've got a growing shoot in a plant which is producing leaves. That will just keep growing until it's told to stop by hormones or other processes within the plant. And one of the reasons it'll stop is when it produces a flower. And when it produces a flower, many plants produce 4 more organs, and then they stop. And those organs are sepal, petal, stamen, carpel. Let's have a look at how that's turned on. This is with some genetic switches. You don't need to know the particular details of it. But if we look on the right-hand side, you see we've got a stalk with 4 buds on it, 4 primordia on it. If we didn't have anything turned on, these would develop into leaves, but we're using switches to turn the leaves into something else. Now, you might think we'd have one switch for sepals and one switch for petals and one switch for stamens and so on, but we don't have that. We have interactions between them. And that's what that little brick wall on the right-hand side is. There's 3 switches, which I've called A, well, I didn't call them. Other people have called A, B, and C. When A is turned on its own, the primordium will develop into sepals. When A and B together are turned on, it'll develop into petals. When B and C together are turned on, we'll get stamens. And when C on its own is turned on, we get carpel. So that's how we use these 3 switches to generate 4 different things. And we know this because we can mutate plants to turn things off, and we start getting weird flowers which have sepals, then petals, then petals, then sepals in the middle, and no stamens or female bits. But you'll know some of this already, because you'll know about double flowers. So how are double flowers made? This is where you've got lots of petals within one flower. And what we've done here, instead of having the A, B, and C exactly like this brick wall here, we can change it, or the expression domains of them within the plant, the developing plant, have changed. And in fact, A is pushed up a bit higher, and C is pushed down a bit lower. And what we get is, instead of sepals, petals, stamens, carpel, we get 2 places where A and B are turned on together. And that means that what were going to develop into stamens develop into petals instead. If you have a good look at a double flower, you'll see very few or no stamens at all. And that makes it a bit of a pain for insects which come there expecting pollen, because there isn't any. It also means they don't tend to set seed. There's an interesting question there about how we get seed for double flowers. I will leave that as an exercise to the reader. Okay, moving on. We're going to run slightly over an hour, for which apologies, but there's just so much more exciting things to talk about. I want to talk about pollination. Pollen contains sperm. The egg is at the base of the carpel, remember? So there's, how do we get that sperm there? What happens is the pollen lands on top of the stigma, and there's a complicated handshake between the 2 of them, which is how the stigma and pollen recognize each other as being of the same species. And if they are of the same species, the pollen starts to rehydrate and pushes out a little tube through one of the slits within the pollen. Here's some lily pollen with a tube in it. That tube has a sperm in it at the bottom, or 2 sperm in it at the bottom, and it gradually grows, pushing its way through the cells of the style, eventually reaching the ovary and bursting. Here's some pollen grains which are highlighted in a fluorescent colour, and you can see, the stigma here is on its side, and you can see them growing all the way through. They can grow really quite fast, up to about a centimetre an hour in maize, but then they do have to travel a very long way in maize. And eventually it gets to the ovule, the pollen tube bursts and releases the sperm. That pollen tube is fed by the cells of the style, by the female cells. And that's how self-pollination works. The female can recognize whether it's pollen from the same genotype as her. It can recognize a signal within the developing pollen grain and compare it to her own. And if they're the same, it's like having a 2-component bomb. It will stop the growth of the pollen tube. If they're not the same, that bomb doesn't go off and the pollen tube can grow all the way down. So that's the basis of self-pollination. So in the last few minutes, I want to look at seeds. Seed sellers want uniformity and beneficial traits. They want beneficial traits because that's what we as consumers want to buy. We want lovely, tasty plants and things like that. And they want uniformity because they want to be able to say, if you grow this one, it will give you give you fruit which looks like this. And those 2 traits don't always, those 2 things, beneficial traits and uniformity, don't always go together. Because breeding true is an important characteristic which gardeners like. You want to be able to plant some beans, grow them, take the beans, plant them again, and have the same thing come up next year. How's that done? What seed sellers do is they generate what are called inbred lines. Here is a cell. It contains 2 copies of a chromosome, which are coloured red and blue here. And when that cell makes a sperm or it makes eggs, information from those 2 chromosomes is shuffled together, and the pollen that it makes, the sperm that it makes, or the egg that it makes, contains some information from each of those 2 chromosomes. And this is what happens with us or any other animal that reproduces like we do, is that you get this mixing of some of mum's DNA, some of dad's DNA, and that's why you inherit traits from both of your parents. But what seed breeders will do is take the pollen and the egg from one plant and cross them together. So we've got shuffled genetic information, but only from one parent. And then we allow it to do it again. And the information is being spread at a slightly finer level. If we keep doing that over generations, what we end up with is an organism where both chromosomes are essentially identical. So it doesn't matter which chromosome you're going to inherit, it's always going to be the same genetic information. That creates uniformity because we know that whatever seed we get from it is going to develop into a plant which looks exactly like its parent. But these inbred lines, they're great for consistency, but they may have some bad traits which make it less good than it could be. And that's where F1 hybrids come in. An F1 hybrid is where we take two inbred lines and we cross them together. So we get, in this case, I've got two inbred lines, one which is purple, one which is green. They make pollen and eggs, and we can cross those together, and the various different plants that we can get out of that, we've got one which has purple from mum, green from dad. We've got one which has purple from dad and green from mum. Then we've got both from dad or both from mum. But what we want is this difference. We want a green and purple. We don't want double purple. We don't want double green. How do we know we're getting that? And there's a really clever thing we can do to make one of those parents stop producing pollen. And that means that we can strip out the double purples, and then we only collect, so that plant will only make eggs. And that means that we can harvest the seed from that plant, knowing that the egg information came from purple and the pollen information must have come from green because there wasn't any purple pollen around. In practice, the way this is done in a field is by having the male variety, which are coloured yellow here, sorry, the male and female variety coloured yellow here, that's the pollen donor, and then the green ones are female only. So that's the ones we harvest the seed from. And they are guaranteed, when they set seed, to have the egg information from mum and the pollen from the other one as well. Now, this is why F1 hybrids are expensive. It's a complicated thing to explain. It's a complicated thing to do in the field. You've got to grow these two different varieties. Sometimes you've got to go there and start taking pollen off manually. It's why F1 seed is so expensive. But when you get two chromosomes from inbred lines coming together, we get what's called hybrid vigor. The child is much more vigorous and productive than either of its parents. And that's why it's such a benefit to gardeners as well. But it's also why F1 hybrids don't breed true. You'll have noticed if you plant an F1 hybrid and then save the seed of it. And it's because we get that shuffling of information. The pollen and the eggs will have, in this case, the purple and the green shuffled together. And then if we allow them to self-pollinate and form a new plant, then the genetics of that plant will not be the same as the genetics of its parent. And usually, always, but usually, those progeny are not as good as the parent it came from. Okay, last couple of minutes, I just want to talk about where crops came from in the first place. Crops come from the domestication of wild species. So back when I said we had our farmer 15,000 years ago or whatever, who was setting up agriculture, they were looking around and saying, well, that plant there, I can eat that plant. I will bring it back and put it into my field. We're domesticating the plant from the wild. And this is where bread wheat came from. About 500,000 years ago, a species of grass called einkorn crossed with another species of grass called goat grass. And that formed emmer, E-M-M-E-R. That's the wheat in the middle. Emmer's all right. It's a bit of a pain to process. It's not terribly nutritionally brilliant. It's still sold in Italy under the name farro. It's one of the three grains which are called farro there. But then about 9,000 years ago, when a farmer was growing some emmer in his or her field, there was a chance cross with another goat grass. And the farmer noticed that it was a taller, bigger, more healthy-looking plant. And actually this cross between emmer and this new goat grass combined the qualities of the two. It was there was a lot of nutrition in it. It was also very tolerant of drought and disease and high light. It was basically indestructible. And so the farmer kept that seed, sowed some more of it, and that's the start of the spread of bread wheat from a chance event about 9,000 years ago. And we are now slaves to this plant. It's actually using us to propagate its own genome, and it's been phenomenally successful at doing that. If we think about it, actually wheat is probably way more successful than we are as a species. And it's really weird to think that this is possibly the most significant evolutionary event since plants moved on to land, because it's been responsible for the invasion of humans on the planet. It has given us that the calories that we need and the ability to grow it and everything like that, that has led to spreading of humans all the way around the planet. And yeah, because of that, it may end up being responsible for many extinctions as well. So it's a really interesting thing to think of, this single chance event with plants having such a big impact. So what's Cambridge doing about food security? We've got loads of different departments working on it. Plant sciences, the Crop Science Centre, that's new up on Histon Road, the Sainsbury Laboratory studying development, the Botanic Garden collecting plants and preserving them, preserving biodiversity, the National Institute of Agricultural Botany, again up on Histon Road, the Department of Biochemistry working on algae and photovoltaic cells and how to get oil out of algae, the Gatsby Foundation training new generations of plant scientists. So I hope what this talk has done has given you an idea of some of the science behind some of the gardening. And I hope it might make you think, when you're going about your gardening, when you're doing stuff which you've always done, just have a bit of a think about why things happen. And the fun stuff comes, for me, in science, not when I do something and it works, hooray, or when I do something and it doesn't work, boo, but when we do something and it, what? Why has it done that? That's the fun stuff, and I hope that you might get some of that. Why has it done that? Go and have a Google of it, because often these things are explained somewhere else as well. And I hope it makes you start questioning some of the stuff you do. Not because you shouldn't be doing it necessarily, but thinking about why it happens. And that's what I find fun as a scientist. And that means it's time for me to go back to my allotment and try and grow some more beetroot, which I'm never very successful. So thank you very much indeed for listening. We have about 10 minutes for questions before I am booked in at the restaurant downstairs. So there is a hard time limit on this one. Let's go.
Could you please flip back to the slide of the channels of...
Where am I looking?
So very at the start, at the channels of... maybe a bit... Yeah, here. Okay. Can I go point it?
Yeah.
So is it possible to use something to manipulate the oxidants to prevent them from going down, so the plant thinks that the spurs have been cut off, so it grows new branches?
Okay, so the question is it possible to manipulate how the channels work so the plant starts branching out without having to cut off the top of it? It may well be. We may be able to produce plants which don't produce auxin in their growing tip or something like that. I don't know of any work which has done that. The problem with it is that auxin is ubiquitous. It does so many things in plants that if we fiddle around with it doing that, then we'll probably break something else. But yeah, I don't see any reason why not. Auxin development has been studied. We're beginning to get to understand it. It's been about 50 or 60 years worth of work so far. So maybe another couple of hundred years we'll have worked out a few more things about it. Yeah.
When the pollen is growing down the stamen, where does it get the energy from to grow so quickly? If it's such a tiny thing, it's way smaller than a seed, right?
Yeah, it's really, really cool. So the pollen grain sits on the top of the stigma, and then the pressure forces out the start of this pollen tube, and it's getting that nutrition from the female tissue. So the female tissue is feeding the pollen tube and providing it with the building blocks it needs to grow that tube all the way down. And this is why I say that it's it's a triumph of feminism, basically. The female gets the choice of which sperm fertilizes her. She is able to say, I don't want a sperm with this genotype, and not feed it. And that's the basis of the self-pollination there. It's not possible for animals to do that, because in animal fertilization, topologically, in a female animal, the egg is exposed to the outside world, if you think about it slightly just more detail than you care to. But within the plant, it actually has to go down through the tissue. It's internal fertilization. So that's where that comes from. Okay, another one at the back.
You mentioned that some plants produce weed killers in their leaves. How long does it take to degrade once the leaves fall down? Because leaves themselves, they take a long time. And I'm thinking in terms of composting, would it be a bad idea then to use leaves in your compost or in your gardening? That's
a very good point. I think I'm going to go for the cop-out answer of it really depends on the plant. And so I think coffee is relatively short-lived. I think it drops leaves relatively regularly because it's a tropical plant. I don't know of any particular plants that you should avoid composting and spreading on your land. I know it's things like plants which have had weed killers applied to them you shouldn't do that with. But I would go for whatever, if somebody else does it and it's okay, then that's fine. Sorry, it's a horrible cop-out answer, kind of hiding the fact I'm not really sure. Any more? Got one on the back. We'll come. I promise we'll come to you next.
You mentioned mycorrhizal symbiosis.
Yes.
And the fact that brassicas don't have any fungi associated with them.
No.
Do we know why that is? Because every other vegetable seems to have something associated.
True. It's just that they never went down that evolutionary pathway. For whatever reason, we're not entirely sure, brassicas just can't do it. They lack the genes to set that up. I talk about this sort of thing with my first-year students sometimes. Evolution is, we always want to consider it in the context of where do we want to get to? We can see that as human. We can see, I want to get there. We can plan. Evolution can't plan. All it can do is incremental steps to make something slightly better than the generation before it. So maybe in brassicas, I'm not sure about this, I would need to go and have a look, but maybe in brassicas, an incremental step which made a child better than its parent was to ditch a bunch of the genes which could have become precursors to that handshaking movement. And then once they've gone, evolving them back is really, really hard. So maybe it's just gone down an evolutionary pathway which it can't get back from easily. And sometimes we need to look at not why doesn't it do it, but the question is more, what was the start of the pathway that stopped it? So we need to go back in evolutionary time and start looking at things there. Again, it's a slight cop-out answer of I don't really know, but this is where I would start looking. Okay, but one more question down the front. I think after this one that will be it. Because I really need a gin and tonic.
You talked about most plants have these root nodules that can get nitrogen
Legumes
from
do,
the
yeah.
air. Oh, okay. Maybe my question is, okay. So what's the point of putting nitrogen in fertilizer and putting it on those plants? Because there's so much nitrogen in the air already.
There is.
Why would you, surely you're going to overdose
Over-nitrogen your things.
Yeah.
So nitrogen is still useful in those cases. The root nodules help the plant, and if we want to help it even more, we can give it more of it, because the roots will still take up nitrogen even when they don't have those root nodule forming bacteria around in the soil to welcome into them. It'll be a nitrate or something like that, yeah. But and the plant can happily take up nitrate from the soil as well. Yeah. Cool. It's time for dinner. Thank you very much indeed for coming. I should also say, I think this is one of the very last events of the Cambridge Festival. They have done a fabulous job with organising wonderful things. Can we just give them a round of applause as well? Oh, that's an interesting one. That's a very interesting question. I think.
Thank you. He's got a rocket. Yeah, he's got a rocket. Yeah.